I'm delighted to introduce Deb McGuinness, who's a senior Constellation professor at Rensselaer Polytechnic Institute. To remind you, this program has been a partnership. Both our program and this SINAInnovations program represents an exponent of a wonderful partnership between Mount Sinai and RPI that really is expanding the use and importance of engineering in medicine. So, Deb?
Thanks very much. It's my honor and privilege to introduce a friend and colleague of mine, Marty Tanenbaum. He's the founder and chairman of Cancer Commons, which is a nonprofit network of cancer patients aimed at improving outcomes using precision oncology. He's a computer scientist, internet pioneer, and cancer survivor. I knew him first as a pioneer in artificial intelligence, leading research divisions and bringing artificial intelligence into practice. But since then, he's led a number of technology businesses, including enterprise integration technologies, CommerceNet, and VioSystems. He's got honors ranging from being a fellow of the Association for the Advancement of Artificial Intelligence to the Melanoma Research Foundation's Legends for the Cure Humanitarian Award. He holds bachelor's and master's degrees in electrical engineering from MIT and a PhD from Stanford. And with that, I'd like to introduce Marty.
Good morning. In keeping with the theme of this year's conference, I'd like to talk about beating cancer from my rather unique perspective, as it started out actually as an electrical engineer before there was computer science, and then computer science, AI, internet entrepreneur, and finally cancer survivor. Where's my clicker? 17 years ago, this month, I was diagnosed with metastatic melanoma, metastatic to liver. And it was a wicked disease with a dire prognosis, basically 9 months median survival. And I was just thrown for a loop. This was at the height of the .com era, and I was successful at it. And suddenly I was looking at not making the millennium. And I did all the research I could do and found nothing that would give me hope. And so I went out and talked to all the local oncologists and then a number of them on the national scene. And no one could give me much comfort in terms of the prognosis. But everyone was saying if you had one shot, this is what I would do. And it was all different. And there was no data to support why one or the other was better for me. And I vowed at that point that if somehow I got through my disease, I had a new purpose in life, because all of my experience as a computer scientist and an internet entrepreneur prepared me to be able to answer that question based on data. Ultimately, I bet my life on a clinical trial that failed. But it did help some people, and I was apparently one of them, fortunate enough to be one of them. And to this day, no one knows why some of us did well and others didn't. And that was enough to get me to redouble my vow. Every person who hears you have cancer goes through a similar life change, where instantly, everything that you were thinking was important in life is out the window. And you're forced to make life and death decisions under the gun, with none of the information and data that you need to make them wisely. You often come home after diagnosis and go to Dr. Google, and you get results like this that say 1.7 million results in 0.29 seconds. And the overwhelming majority of these will be either irrelevant to you or incomprehensible to you or not clinically actionable or out of date or just wrong. And most importantly, the nuggets of information, the needles in a haystack that you need to be able to determine how to beat this disease are going to be buried somewhere on page 500 or 800 of this thing. And so this is, search engines look for what's popular, not for the needles that are unique, the information that you need for you as a distinct patient. And so this is a problem. So what do patients do? They do the same thing I did. They might not have done it to dozens of people. They'll go out and talk to different oncologists, and they will get conflicting opinions. And they won't have the data to decide either what to do, because the reality is that in this field, there's far too much trial and error, much too little data, and very little learning. Everyone has to start from scratch. When you're doing that Google search, it's not like you can learn what are the good articles that someone else might have found buried deep down. You start from scratch. And this stuff matters. The 5-year outcomes, or whatever other measure of success you want to measure in cancer, vary widely, sometimes as much as 50% as you move across different sites, from one major cancer center to another, depending on how much expertise they have in yours and your cancer. And this stuff falls off very rapidly as you move from a major cancer center out into the community, and then from there out into rural communities or disadvantaged communities or the third world community. And I consider this outrageous in the internet age, that information should be so unevenly distributed. And I want to give you a personal example of this. Dr. Morton, Don Morton, is the guy who saved my life. He's a legendary physician who invented sentinel node biopsies. For those of you who are docs, he was a phenomenal guy. He also had a lifelong fascination with melanoma. And this paper is the one that saved my life. And I never would have found it if I was just Googling. I found it because someone had tipped me off about Dr. Morton, and I went looking for papers that he wrote. And it basically said that for something like 34 patients in a sequential study of 1,750 patients with melanoma metastatic to liver, there was sufficiently low disease burden that he felt comfortable doing surgery. And of those, when he opened them up, 24 of those 34 were actually operable. And of those, 20% had long-term survival. So you want to know what lottery I won? I was like 0.4% of the 1,750 patients. But nonetheless, for patients like me, this paper was a lifesaver that basically said that metastectomy with intent to cure, to take out those mets, followed by a clinical trial, the one that failed, had some hope of working. But back then, maybe no one knew this was the best, but someone knew what would work for me, and I had no way of really connecting to them other than just pure luck. This is a paper that was published posthumously. Don died earlier this year. And it was published a few months ago, looking back at a retrospective study of a few hundred patients like myself. And this says that this stuff actually works for some selected subset of melanoma patients. And it's time to mark a departure in the way melanoma is treated. This is 16 years later. So 16 years ago, someone knew how to treat me. Maybe not everybody, one person at least. Now they're saying this is something everyone should know about. Now, this is published in the Annals of Surgical Oncology. This is not got a lot of, it's not the American Cancer Society. Google won't put this on the front page. The surgeons already know to try to take something out if it can come out, but the medical oncologists, which is where most metastatic patients are seen, will not treat melanoma patients with surgery once they're metastatic. They don't think it can work. So think how many patients are going to die needlessly. The thought that comes to my mind when I see this is, if only we knew what we know, right? If only everybody knew what some people know, then we would save a lot of lives. I think back over the last 16 years, how many people like me might have been saved by following this procedure. And things are going to get a lot worse, because we're now entering an era where cancer is known to be thousands of molecularly distinct subtypes. And it's quite possible that every person's cancer is unique. So when you think about the fact that these diseases are going to be increasingly managed like AIDS, they're going to be treated by combinations of targeted therapies, the sheer combinatorics, thousands of diseases, tens of thousands of combinations of drugs, there's no way that you can use either classical clinical trials or trial and error in an efficient fashion with no learning in order to be able to understand the space. So what can you do? Well, the good news is that we're on the verge of a new era in precision oncology, which is going to transform things. And the basic idea, which you may have picked up during this conference, is that you take a sample of a tumor, send it out for various kinds of omic analysis, find out what is different about that tumor that might be unusual in melanoma but common in some other disease for which there is a treatment, and then try that. And you can get these cross-learnings going. So here's a report, typical report that you get back from one of these tumor screenings from Foundation Medicine, which is one of the leaders in this field. And you'll notice that they've called out a few variants, which are unusual and clinically actionable in this particular specimen, and some FDA drugs and some trials that might be considered. The problem is that these reports raise as many questions as they answer, and probably more so. And in the best places, these reports are just input to molecular tumor boards in which people are looking at the recommendations and saying which of these genes are actually the dangerous ones, which ones are the drivers, which ones are expressed, which of the drugs that are possibly recommended should be used, or trials, and in what order or what combination. So there's a lot of science that goes on. These guys are engineering solutions to these patients in real time. And they're considering all kinds of things, such as what the mutations were, what the clinical history was, what's known about molecular pathways. There's often scientists participating on these boards as well as doctors. And the reality is that virtually no one publishes case reports anymore. And so all of this learning is lost. And that's really a tragedy, because we're not just talking about how the patient was treated and what the result was, but we're talking about all of the treatment hypotheses that were considered and the rationales for rejecting those, because there's a lot of learning there. Every patient is potentially, an experiment, each drug a probe, and we can learn a lot more than we can typically learn from the population statistics that come out of a clinical trial. So all of this background, which includes that there's something will work in a patient that the patient doesn't know about, or something that might work that we don't know whether it will work because we don't have the data, or learning in these N of 1 experiments, which are just lost completely. That was the motivation for my starting Cancer Commons, which is a nonprofit rapid learning and discovery network with the simple mission of giving each patient the information that they need in order to pick the best possible treatments. I would say patients and their physicians, because the patients are not going to treat themselves. Then learning as much as possible from each patient's response and disseminating that information as rapidly as possible. And by that, I'll draw the contrast through the traditional medical literature, which is where most of you will think it's the right place to publish. But if it takes, on the order of 6 months to a year to get something through peer review and published, and then another year for someone to publish comments on it, you can see how patients and the care that patients are getting are going to fall increasingly behind the frontier of knowledge. It just doesn't work anymore. We need something that will do things. So at a 50,000-foot level, here's the way it works. A patient presents. There's a model of molecular subtypes of cancer and how to treat them, the best treatments, the best trials. A patient gets assigned to a treatment that's within that class that's thought to be good. The response is analyzed. And if the patient is not responding as expected, we go right back to the scientists where this information came from and say, what did you guys not understand about the biology of the cancer or the mechanism of actions of the drug? And by the way, you don't have 5 years to do research. You've got 3 weeks. This patient who just failed therapy is coming back into clinic. I want you to work on this as if it was your own kid or your own spouse. That's what we're talking about. Let me zoom down on the model. And this is a particularly simple model. It's one that we started with a couple of years ago for melanoma. And on the left are about a dozen subtypes, which correspond to, they're color-coded, either a mutation or a pair of mutations in one or more of the driving pathways of melanoma. So you have the MAP kinase pathway in yellow and the PIK3 pathway in blue. This one's like a gas pedal. This one's like a brake pedal. Patients get assigned to these subtypes, and eventually some patients will respond and some patients won't. So those pathways will get split into responders and non-responders. And then some of the responders will in fact recur. So this becomes a tree over the space of targets and treatments. And what we need is an efficient way to search that tree. If you want to know how big that tree is, Eric Lander, who was one of the leaders of the Cancer Genome Project and director of the Broad Institute, estimates that there are about 100 operable pathways of interest in cancer, either because they're oncogenic drivers or because they're resistance pathways. And it's just a matter of time. 100's a big number, but it's not a huge number. And it's only a matter of time till we systematically explore that space. But we need data in order to do it, data that's often not widely shared. So Cancer Commons is building a platform to encourage that kind of data sharing and collective analysis. And here's a picture of it. We start with expert curation and big data analytics and crowdsourcing in order to be able to take the world's knowledge of cancer and organize it into public databases. And I emphasize public because if it's not public, we can't get the kind of tremendous multiplier effect of everybody working on it that happened in the Cancer Genome Atlas. Not much happened on Craig Venter's branch of the Human Genome Atlas. This stuff has to be public. And most of these models are models that other people are providing, but I point out the middle one, which is the model of molecular subtypes and how to treat them, which is the model that we think is particularly interesting and the one that we're building to connect the clinic with the research, because molecular subtypes is that connector. And then the various stakeholders interact with these models using applications, which for rapid learning, physicians and patients need applications to help them make treatment decisions, to discuss those decisions with their peers, and to report outcomes. Whereas physicians and scientists, clinical researchers, might need applications to help them analyze the results, discuss those with peers, and then update the models so that we drive this continuous learning process. I would like to focus for a second on the applications that are for patients and physicians, but let's look at patients first. And what we've developed is what I sometimes call the Google Maps of cancer, which is to take all of that world's knowledge and contextualize it and personalize it, so these are the relevant alternatives for a patient being treated for a particular type and stage of cancer. And so you start with the standard of care guidelines, and then you provide the ability to drill down into, to be able to get the information that you need to make tough decisions. And these are the types of things like that reports, those papers from Don Morton, that should have been on my map, 16 years ago, as opposed to all the other things that might have been irrelevant. Or case reports from N of 1 patients who are like me, or relevant research abstracts, or clinical trial abstracts, or news articles, and so forth. What we then do, or firsthand reports, by the way, from patients who are actually on a drug or in a clinical trial, by the way, that might be blinded. And no one knows what's happening, but collectively the patients know. So we're getting early signals out of this that can be used to accelerate the movement of patients in the direction of promising trials. What makes this really exciting, though, is doing the moral equivalent of what Waze does to Google Maps, which is to overlay real-time data based on the clinical results of patients like you. This is not the general population, but what did patients like you try and how did it work? And then we close the loop by asking patients, what did you do and how did it work? And so a virtuous learning cycle emerges. And over time, the branches on this map, the treatment pathways on this map will split. into responders and non-responders, and new trials will appear corresponding to the off-road experiences of pioneers who are trying new kind of N-of-1 treatments. So when a patient is off the map, the important thing is to direct them, not say, go have a clinical trial, but to direct them to the specific things that might best help them so that we can maximize learning. And so, for example, the choices that might exist for a patient who's out of options is to get sent to a clinical trial, but not just any clinical trial, a clinical trial that's specifically optimized to do good for that patient. And so you're looking at, is there a good match between the genomics of the targets on the patient and what the trial's trying to address? Is there a strong scientific hypothesis underlying this trial? It's not an incremental advance. Is it a good scientific design? Is it a patient-friendly design in the sense that patients who are on a control arm can get to cross over if it's not working? So these are very selected trials. Alternatively, we might send a patient to a molecular tumor board, which is practicing state-of-the-art oncology, precision oncology, where you're not just talking about sequencing a tumor for mutations, but you're throwing the 3.5 terabytes of data at them that Andrew was talking about in the previous talk, in order to really deeply understand what's going on, to reverse engineer that tumor, and to engineer rational therapies that perhaps can be tested in vitro or in vivo before you put them into the patient. And then occasionally, you might be able to connect the patient directly with a researcher who's doing something that's relevant that might help them. Maybe the researcher is even doing research on cell lines that originated from cells donated by that patient. But in today's system, the researcher will almost never know who the patient is. Researchers say it's coming up lucky, he's knocking things out of the park, it's working in a mouse model even, and the patient's dying. Just think how many, we could slash a decade off the time it would take to get this treatment ethically into the patient if we could only connect them. And we're not talking about does the patient live or die. You know, in 6 weeks, we'll get some objective early indication of whether this drug is working. And if not, the drug will be a probe. We'll learn something and we'll improve. And if all else fails, we are turning to crowdsourcing. A colleague of mine, Tony Blau at the University of Washington, is putting difficult cases up on the web with all of the data, information. And more often than not, these cases are getting responses that are helpful to that patient. And while I don't have time to go through the crowdsourcing in detail, I will say the biggest observation is that if it's, say, a breast cancer patient, and you have all these breast cancer experts, that someone, and in one case it was me, I didn't know anything about breast cancer, but a melanoma expert, sort of expert, was able to recognize what was going on in an inflammatory breast cancer patient whose cells had rash all over their skin, very painful. And the rash was inflammatory cells surrounding cancer cells, and they were standing off. And to a melanoma patient, it's kind of obvious that maybe there were cytokines being emitted that were causing the inflammatory cells to stand off, and maybe the anti-PD-1 drugs that were done just approved for melanoma about a month or two ago might be something worth trying, and that is something that I hope will be tried. So the important thing is to learn everything you can from these experiments and get them on the map in less than years, right? So that things that are successful can get rapidly replicated and help other patients, and things that are not successful can, as they say, fail quickly and not chew up more patients. In order to be able to expand the use of these N-of-1 studies and be able to provide expertise across the field, because not everyone can be experts in everything, and in order to be able to coordinate the search over that search space of leads and targets, those red branching things that I showed you earlier, we're forming a network of like-minded people who are interested in doing this. And I would like to point with pride to our newest members, Eric Schatt and Joel Dudley from Mount Sinai. And we're looking for a very close partnership with these fellows. But the goal is to make sure that as many people as possible are able to take part in this, and that we're able ultimately to coordinate these kind of N-of-1 experiments so that everyone doesn't get the same thing. right? We want to be able to balance the utilities of current patients against the discounted utilities of future patients in order to be able to efficiently explore all promising alternatives. Our goal, 5-year goal, is to cut cancer deaths in half, not by curing cancer, not by curing the other half that I will admit we don't know what to do with or do for, but to be able to help the half of patients who are dying unnecessarily because they don't have access to what everybody knows. And given the fact that the wide variation that exists today in cancer outcomes, and the fact that we can get a long ways by not inventing any new knowledge, but just making sure that people are matched to the information that they need to know, I'm quite confident that this goal is achievable, perhaps if not in 5 years, certainly in 10 years. But the challenges are not the technological challenges of information technology. They're all of the socioeconomic, legal, regulatory challenges that are involved in, for example, getting the cancer culture to understand the importance of being able to share data on treatments and outcomes so that we can make some sense of all of this genomic knowledge that sometimes is being shared through government programs. Or being able to convince pharma companies that when there's a strong rationale in an N-of-1 study for an off-label use of a drug, they should provide that drug, and they'll stand to learn a lot from it. Or being able to get reimbursement for those drugs when they do work, contingent reimbursement, and getting the FDA to have a regulatory policy which encourages early experimentation with combination drugs. So in the few minutes that remain, I'm going to talk about data sharing, which is the one thing that I know I can do something about. And we're trying to set up a network of partners who have expressed a willingness to, in exchange for us referring patients who are needing extra help to them, they provide back the data on those patients, clinical data, genomic data, whatever it is that they have. And in exchange, what they get back is aggregated data, which they can use for all kinds of purposes to improve outcomes, say through variation reduction or comparative benchmarking. If they're pharma companies, they can use that data to be able to see early signals that a drug is working, or be able to recruit the right stratified patients who are ready to very rapidly replicate things and move on. For patients who are patients at institutions that are not affiliated, we're going to work with those patients to get them to exercise their HIPAA rights, to get the data out and donate it to the public. But it's important to be able to work with institutions so that we can get this information without undue delay and get it properly validated. So I would like to summarize by saying first what it means to beat cancer from an engineering perspective. We'll start with the war on cancer, which has very mixed results, and people are not happy given how many billions of dollars have been spent with what's been achieved. But it's possibly because we're looking at the wrong goal. The war on cancer is a monolithic war against a fairly monolithic enemy, whereas we now recognize that cancer is thousands of diseases, and we're trying to treat them at the large scale of the war as if they were a single disease. It makes much more sense to be able to manage an individual's cancer, learn as much as you can from that terabyte of data, and then rapidly replicate and spread that learning, doing it with the kind of loop that I have talked about, where you provide information and then learn as much as possible, disseminate widely. And by tightly coupling research and care, I think we can both improve outcomes significantly and significantly accelerate research. Cancer Commons' role in this is, first of all, to be able to have this goal of reducing cancer deaths by 50%, starting with getting rid of the unwanted variation. And what that means is take this map and put on the map the things that we know that work. So this is not Don Morton 16 years ago, because we didn't know if it worked. This is Don Morton 16 years now that we have the retrospective study. And if someone has melanoma metastatic to their liver with small tumor burden, there's a way to treat this, and the patients ought to know about it right away. 16 years ago, Don Morton was one of many things that might have worked, and we just needed the data. So the second thing we need to do is capture the data from every patient, treatments and outcomes, in order to understand what works and what doesn't work. And then, most importantly, for the patients who are off the map, we have to make sure that these patients are getting access to the best science, that science is not reserved for treating lab rats, but we're going deeply and studying what's necessary in order to treat those patients. We have to be able to capture everything that's learned about those, from those experiments. We have to make sure that as many people as possible are getting access to that kind of service, and we need to coordinate those studies in order to be able to efficiently study the search. So I will conclude at this point by just taking it back to the patients. How many of you have been personally touched by cancer? Show of hands. By the time your life journey is over, everyone in this room will have their hand raised. Half of you, roughly, as patients, a third of the people who, the others, family members, third of people who get cancer are going to die from it. It doesn't need to be that way if only we knew what we know. And so I'm going to urge you in my final sentence to do what you can to open access to your data, to share your results, to open access to your code, to your expertise, perhaps in biocomputing, so that we can work together to beat this damn disease. Thank you so much.